Well, one of the things that you collaborated on was a project called DynaCLR. There was a paper that I think came out last fall titled, Contrastive Learning of Cell State Dynamics in Response to Perturbations. In reading that paper, my sort of sense of it is that, ultimately there's a problem that we're trying to solve, which is that when, cells are exposed to perturbations, a la viruses or maybe molecules and so forth, kind of understanding how that process unfolds right now is a very manual, kind of tedious process. It can be error-prone if it's done right by individual scientists, depending on whether or not they had their coffee this morning, right, or maybe how they got trained or their facilities. And it seems like you've developed this machine learning model that helps to sort of automate that in some way using some specific techniques in AI or ML. So maybe big picture, am I getting this right in terms of the problem that you're trying to solve here?
Yeah, I would say the high level problem is really, if you think about like therapeutic design overall, it sort of relies on understanding how different perturbations like gene knockouts or drugs are changing the dynamics of single cells. But traditionally, the ways to do that is like you use sort of static imaging techniques, or you have gene expression assays that are developed. But that really provides a more static snapshot of the state that the cell is in. And so if you perturb that cell, really understanding the dynamic changes in terms of the phenotypical impact of that, and also the dynamics is a big area. And so the group that I worked with at CZ Biohub was working, like I said, a lot on live imaging. And what's exciting there is you can collect large volumes of data at multiple different channels. So we were working with 5D data across XYZ and then across time and different frames. And so there was 5D data we were collecting across different channels. And so if you think about the impact of that, you can look at fluorescence channels where you can have viral proteins or organelle markers that you can track over time, or you can just look at label-free channels that give you a really good understanding of the physical structure of the cell. But the hard piece there is how do we annotate this data, right? And that's a super manual process that can be very slow and labor intensive. And so the idea there was, can we build a more efficient pipeline to understand that and do some really cool downstream analysis based on those embeddings that we get from these models to then understand how infections, for example, are impacting the cell, or be able to detect rare events in these cells like cell division.
Well, maybe zooming in a little bit. So the paper talked about DynaCLR being a self-supervised learning framework that leverages contrastive learning specifically adapted for analyzing biological time-lapse data of cells and organelles. Maybe just breaking that down a little bit. So, self-supervised learning, again, in my understanding of it, versus supervised learning. In supervised learning, you kind of have the answer key, right? You tell a model to maybe make some predictions, and you can actually tell whether those predictions are right or wrong based off of some existing, already annotated, accurate data set. In self-supervised learning, you're kind of asking the model to do more of the work and kind of guessing itself. So I think one example they give in the worlds of language is, a sentence with a word taken out and has to, figure out what that missing word is, or maybe parts of an image that have been sort of removed and has to fill in that image itself. Can you share more about sort of why self-supervised learning was maybe the way to go for what you were trying to achieve versus other approaches?
Yeah, I would say one of the arguments is obviously you don't have a ton of annotated data, right? And so you don't have ground truths so to say that you can use to train these models and validate the accuracy. But also really there's an argument to be made about using these embeddings that can sort of learn a more general representation of different cell states. And that was really the argument that we were making in the paper is, hey, if we use a specific training objective function, which in our case was using the triplet loss, which is commonly used as one method in contrastive learning, we can actually really, if we structure that in a clever way and adapt that to these really large scale 5D images that we're collecting, that can actually help us then learn a more general representation of understanding different effects of these perturbations.
So you talked about contrastive learning. Can you maybe share more about how do you define contrastive learning?
Yeah, I would say there's different sort of methods. I mean, there's like the SimClear paper, which most people have sort of worked on and iterated on. And so we used sort of a variant on top of that, which I call triplet loss. And essentially, I guess the idea there with our framework was we had, let's say, an anchor pair, which could be just like let's say, an image of a cell, right? And then that can be a cell at a very specific randomly sampled time point, and then you build a positive pair, which is let's say the same cell but at a later time point. And so at that point you've sort of seen the, let's say, infection progress a little bit further, and you're seeing the impact of that. And then let's say your negative pair is just a completely different cell. And the idea there is like within physical space you have these images, and so which images belong to each other and which ones are closer in terms of time, and can we really do a similar parallel mapping within embedding space. And so the way that contrastive learning does that is the goal is to bring together the positive pair closer to the anchor pair within this sort of embedding space, and then the negative pair should be further away from the anchor pair, since that's a different cell. And so using that sort of framework, in our case, that was sort of the argument we were making, and we tried out 3 different sampling techniques that we also describe and run experiments on. One is sort of more of a classical contrastive learning method. We looked at cell-aware sampling. We also looked at cell-aware and time-aware sampling to show how adding that temporal effect can be useful. And a lot of my work was focused on sort of looking at cell division. So we used this open source data set, Alfie, where they have a ton of data and annotated data actually on cell division, and really from the beginning all the way to the end, the cell going through mitosis and whatnot. And so you can really use that as a way to see, is our model learning these rare sort of cell division effects that are really hard to capture, because in terms of data you're collecting, that could be a really short time scale that maybe your microscope isn't capturing. And so that's one area that we found it to be useful. Obviously also looked a ton at like just viral infection, because CZ Biohub is doing a lot of stuff with that. So we looked at Zika, dengue virus. So that's one area. Another area, and then third area was also organelle remodeling. And so we looked at early evidence of that within ER remodeling, but also expanding that to other organelles as well.
When you develop these kinds of embeddings, is the idea that once we've develop those, we can generalize across different cell types and different types of viruses, for example, besides dengue or Zika, or are they really specific to like the kind of cell lines that you've used in these specific types of viruses?
Yeah, so I would say DynaCLR is like more of a framework for what we found in terms of like setting up a training pipeline that really gave us interesting results. And I think using that framework, you can definitely build a foundation model. And we do some of this work as well in the extended preprint, where we expand to different cell types. And Alfie was one part of that effort, because they have a lot of different cell types that they're looking at there. And so initially we looked at A549 cells, which is a specific type of cancerous cell, but you can also expand that into other cell types. And so that's really the argument to be made with embeddings, is that you can generalize to different cell contexts.
So, you mentioned that there's this kind of automated, maybe microscopy component of this that they're working on, and then there's also the, machine learning algorithms that, you were working on and developing. As we look to continue to make progress in this field, I guess, do you find that, more progress is going to be unlocked through just, again, getting access to more data by developing, better and maybe more, kind of these microscopy workflows that are more automated and can generate more data sets? Or is it going to be maybe more on the sort of like ML and kind of math side of things?
I think it's definitely a combination of two, right? And I think they sort of go hand in hand. You need a lot more data for these models to eventually scale and for us to be really confident in the predictions that they're making. And so that will definitely require a lot of innovation on the hardware side. But I do think there's a lot that can still be squeezed out from the data we already have on sort of the method side. And so, obviously there's different modalities you can look at. We were looking at microscopy data, but you could also look at gene expression data, and there's a lot of stuff happening around just multiomics and using different types of techniques and merging them to draw greater insights. And so I think there's the scaling of data, but also just using like a multimodal approach that can help unlock a lot more. And then there's sort of a methods side of it, which is just how do we use the data we already have and extract more from it.
Very cool. Well, I imagine that as you are, developing, it's called DynaCLR. Is that how you pronounce? Okay, not DynaCLR. Corrected. So
I mean, you can say it either way.
Okay.
Yeah.
DynaCLR sounds a lot cooler, I think.
Okay.
So, as you were working on it, I imagine, some of the challenges that you might have been dealing with are just, for example, the volume of data and of images, right? And being able to, work with them in ways that are computationally, efficient and so forth. There might be sort of variability and noise within some of the data that you're looking at. So, I don't know, was there a specific sort of, interesting challenge that you were involved in or saw the team, really have to overcome in order to make this work?
Yeah, I worked closely with Zuen and Ed, who are part of the team, and I'd say the biggest part sort of in terms of processing the data in an efficient way was just, one, you have terabytes of data. So I think the hardest part is really just how do we get this data to load efficiently. And so I think there is a lot of pipelining work that went into that, so making it just generally more efficient. But also I think there's a lot of stuff within the actual data collection and just the pre-processing that goes into registering the images and making sure that if you go in and it's not like you just collect the data and train these models, right? There's a lot of, I think, human just inspection that also goes into making sure what you're seeing is validated with the actual data. And so predictions our model would make, we actually went in and we would see, hey, looking at specific cells and data frames, are we actually seeing similar results with the work that we did? And one thing that I think helped with that, we had a specific sort of infection sensor that we had built. And so that was really helpful for just having a GFP reporter that would basically translocate into the nucleus. And so if you have, microscopy, you can see that under sort of a fluorescence microscopy and see, hey, the cell is actually infected now. And so we would see, different patterns for infected versus uninfected versus dead cells. And so, even just using the human eye, you could see, hey, what's going on in this cell. I think that was a really useful way to sort of make sure the results we're seeing make sense. But yeah, I would say there's definitely the processing side of it, but then also a ton of just validating the results and actually spending time closely looking at the data.